Hello, and welcome to HTAI's annual virtual meeting. We are excited to present What Does Good Look Like? Evaluating Decision Grade RWE, Part 1. As we wait a few minutes for further participants to join, I will share a few slides with you about this webinar. Participants throughout the webinar will remain muted. To submit your comments or questions, please use the questions function in your GoToWebinar control panel located on the right-hand side of your screen. Thank you again for joining us. We hope you enjoy today's session.
Welcome everyone to our workshop, What Does Good Look Like? Evaluating Decision Grade Real World Evidence. My name is Ashley Jacksa, and I'm going to be one of the presenters today, and I'm joined by Nicole Gatto, who is also going to be one of our presenters. If we jump to the next slide, we can introduce ourselves a little bit more formally. I'm Ashley Jacksa again. I'm the VP of Analytical Solutions at Aetion. And my role is really to focus on partnerships with regulatory and HTA agencies to help them understand real world evidence and to help them design the methodological guidances for how they will evaluate and use RWE in their decision making. Liz Gary, who will be joining us for the Q&A and for the next session where we do some hands-on activities, she is a principal scientist at Aetion. And prior to joining Aetion, she managed an AHRQ-sponsored National Resource Center for Comparative Effectiveness Research. And she has also worked with Brigham and Women's as well as Boston Children's Hospital. And Nicole Gatto, who is joining me today, she's Senior Vice President of Science here at Aetion. And her role is really to focus on our COVID work with the FDA, as well as working with our clients on submissions to regulatory bodies. And she leads our internal research here at Aetion. And before Aetion, she was the Epidemiology Group Head at Pfizer. And we are going to be talking to you today about real world evidence and how to judge the quality of that evidence. You jump to the next slide, Nicole. Just a quick look at our disclosures. We are all Aetion employees, and we have an ownership stake. We are also going to be, this is more for next session, but we're going to be using the Aetion evidence platform to show you how to generate real world evidence. And this is, while we'll be using that platform for the analysis, it's no endorsement of a commercial product. In terms of our agenda today, if you go to the next slide, really we're going to focus on what makes RWE decision grade. So we're going to do a quick overview of what is real world data versus real world evidence and how it's used in HTA. And then we're going to look at the status of HTA guidance, RWE guidance from regulatory and HTA bodies. And then Nicole is going to do a detailed discussion on the best practices for RWE generation and evaluation. And then we'll do a quick preview of part 2, where we will we'll discuss how we're going to choose our own RWE adventure, which will be part 2 tomorrow. If we go to the next slide, we can jump in to a quick overview of what is real world data and real world evidence. Go to the next slide. We want to just level set on some definitions here. So real world data is data related to patient health status and or the delivery of healthcare routinely collected from EHRs, claims, registries, patient reported outcomes, and devices. And real world evidence is the clinical evidence about the usage and potential benefits or risks of a medical product derived from analysis of that real world data. So we wanted to provide this as just a level set. That definition was from the FDA. So what is really compelling about real world evidence? Why is there so much hoopla around its usage right now? So real-world evidence allows us the opportunity to ask additional questions, more questions than we typically ask in randomized controlled trials. And it gives us a broader understanding of different populations, and it allows us to generate more contextual evidence that is really, that could be feasible to do within the randomized controlled trials. So for example, if we think about populations used in in randomized controlled trials, it's typically more healthy folks. In RWE could allow for the study of patients that have more comorbidities, or for example, maybe someone that is pregnant, which would be excluded from a clinical trial. So in terms of the opportunity for the use of real-world evidence in HTAs, we think it's vast. We think that RWE can help both with the assessment design, so what topics are of interest, what topics are of importance within a particular jurisdiction, as well as the scope of the assessment. So what patient populations are relevant, what comparators are relevant, what outcomes are relevant to that specific geography or jurisdiction. And not only just helping with the design, it can also help to fill the efficacy effectiveness gap. So that's things like clinical effectiveness, how the drug is actually performing in the real world, as opposed to how it performed in the randomized controlled trials. Cost effectiveness in terms of increased kind of specificity of inputs to the cost effectiveness models. And of course, what we would call landscape management. So this idea that over time that the drug is marketed, we have the ability to accumulate real world data, and you can start to understand if the drug is living up to its potential that was, the potential for cost effectiveness and efficacy that was discussed or that was reviewed or evaluated in the initial assessment. So HTAs have used real-world evidence. Real-world evidence is not new. Observational studies have been around for a while. They've been used in evidence-based medicine for a while. And we do see historical use of RWE in HTAs. And most often it is used for cost-effectiveness modeling inputs. So that's understanding, prevalence and incidence of disease, understanding the costs associated with treatments and with the side effects of treatments and so forth. And it's rarely been used, though, for comparative effectiveness research. And so this is where we are focusing today. We're focusing on, how do you judge the quality of RWE and comparative effectiveness? So there are historical challenges as to why it hasn't been used for comparative effectiveness. And one of those is the idea of evidence hierarchies, which typically place randomized controlled trials at the pinnacle and everything else, like observational studies, lower down the ladder. And of course, there's also a lack of clarity around RWE methodology and how RWE can be used to evaluate causation. And there's some concerns around the transparency and trust within the methodology and within the RWE. So this idea of a black box, that RWE studies are more opaque. We jump to the next slide. We can start to see that this is starting to change. So we see a number of HTA agencies that are contemplating how they're going to incorporate RWE in their assessments. So for example, we have NICE, who is just released a statement of intent to utilize RWE in their assessments, so not just HTA, but all of the guidance that they're creating. And they're currently working on their methodology standards for how they're going to generate and incorporate RWE. We have other agencies like IQWiG in Germany, who have recently announced that they're accepting high quality RWE that is generated from registry data. And they believe that registry data, and not claims or not EHR, but registry data only, really has the opportunity to meet the requirements for the benefit assessments. So while IQWiG is becoming a little bit more open to RWE, there's still some additional steps that they can take. But this is good news. This is a push in the right direction. And then we see ICER here in the US, who announced that in 2020, they're going to be generating RWE for their assessments. And their idea here is that they're going to use this RWE to, one, fill some evidence gaps that were discovered through their systematic review process of a particular therapy, as well as they're going to be using RWE for reassessments, so looking at a drug 24 months after marketing authorization. So these are three agencies that we went a little bit more in depth in, but if we go to the next slide, the next two slides, really, we can start to see that it isn't just ICWG, NICE, and ICER. We see other agencies moving more towards the green here, more towards the openness to RWE. Let me jump to the next slide. And then we can jump to slide 13. So we know that HTAs are signaling that they're open to RWE. So that really kind of begs the question, what type of guidance have they issued to determine or to define what is high quality RWE? So what we did is we looked at seminal publications from HTAs, regulators, payers, and other stakeholders like ISPOR, HTAI, ICBE, to really evaluate how these different groups are defining what is high quality and how they're planning on using it in their decision making. And what we found was a lack of prescriptive guidance. So these groups haven't issued a comprehensive targeted recommendations for what high quality RWE is and how they're going to use it. Instead, we have a patchwork of recommendations on what we think are the key building blocks of RWE. So we created a framework to really organize these position pieces to start to tease out where we are on the road to prescriptive guidance. So the next slide shows our framework. And really, again, the end goal here is for researchers and manufacturers to really have a clear and comprehensive understanding of how decision makers are defining high quality RWE, how they're going to evaluate that RWE, and how it's going to be used in their decision making. And we're calling this prescriptive guidance, and you can see that over here on the right. And we're not there yet. So we don't see too much activity in terms of prescriptive guidance. Instead, we have this patchwork of position pieces, recommendations, and so forth on their commitment to use RWE, which is over on the far left. And then, of course, what we would consider the building blocks of RWE. And the building blocks really follow what we think are the key components of an RWE study. So from hypothesis generation, which is kind of taking into account where it is RWE useful, to study design, data source considerations, analytical methods, transparency, and reproducibility. And what we did is we organized those seminal publications into this framework. If we flip to the next slide, you can see the smattering of the different publications listed underneath each kind of building block. And we see that some areas have a lot of activity while other areas do not. And really the point here is that we don't have this prescriptive guidance, but we are making headway. So the FDA is obviously, you probably are aware that the FDA is working on real world evidence guidance, and it's expected to be published in 2021. And we've already discussed how NICE is already in the process of working on their methodology, recommendations, and guidance. So when Nicole starts to explain what we think of as best practices for RWE generation, this is really taking into account this collection of activity and recommendations, position pieces, and so forth that have been put out by agencies and other stakeholders, but it doesn't necessarily reflect just one agency's guidance. So we're going to turn now to Nicole, who's going to then go through what we would consider best practices, taking into account, like I said, all of these different recommendations from key stakeholders. Nicole?
Thanks, Ashley. So I understand that most of the people in the audience today are more likely to be evaluating the quality of a real-world evidence study as opposed to designing and implementing a real-world evidence study. And going back to Ashley's last slide, in terms of these building blocks, the final report evaluation box on the close to the right-hand side is the most relevant. But we believe that the best way to use these checklists and these recommendations to evaluate the quality of real-world evidence is to understand how good real-world evidence should be generated. And so that's what we're going to walk through in the next section. So as you know, there is a spectrum of research approaches. So on the far left-hand side of this continuum, we have highly controlled experimental research. And on the right-hand side, we have real-world observational research. And the highly controlled research, we're typically talking about a randomized blinded controlled trial. At times it could be triple blinded. These tend to be the most valid, but also have the most operational requirements, sometimes pose risks to patients, and they're the least real world and least efficient. On the far right-hand side, we have the secondary data epidemiology studies. So in these studies, we're using data for a secondary purpose. The data were not collected for research. And for this reason, it's really the most real world. You can think of these as you as the researcher are a fly on the wall observing how behavior of physicians and patients happens in the real world. So the most real world, often the most efficient, doesn't really pose any additional risk to patients. And but for the reasons that I mentioned, that these are not data collected for research purposes, validity can be difficult to achieve in the study design. And then, of course, we have studies along the continuum in between. We have pragmatic trials, we have primary data epidemiology studies that have some non-standard of care assessments. We have primary data epidemiology studies that have only standard of care assessments. And within this continuum, there are also sort of layered continuum. You can see a layered continuum. So an example here would be the randomized pragmatic trials. So the version that would be closest to a randomized trial here is taking a randomized blinded control trial and taking one of the controls away to make it more pragmatic. But you can see that on one end of the spectrum, and all the way on the other end of the spectrum are large sample trials, which are essentially observational cohort studies that start with baseline randomization and then follow people observationally throughout for the rest of the study. And that could be through primary data collection or secondary data collection. So in some cases, a large sample trial might be even more reflective of routine care than some primary data epidemiology studies. We know that many research questions, particularly around post-approval safety or effectiveness, may pertain to one or more real-world conditions. So we may be interested in understanding, once a drug is on the market, is the drug still safe and effective when patients are exposed for longer than studied in the trial? When we're looking at larger populations, maybe there are rare risks that weren't seen in the trial. We could also be looking at more diverse patient populations. People are taking concomitant medications that weren't allowed in the trials. What happens when the prescribing physician is not a specialist in that field necessarily, when there's off-label use or non-compliance. And sometimes when testing that is required by, even in the product label, is not standardized as it was in the protocol for the randomized trial. And then also when patients are just seen less frequently in the real world. But we can go beyond thinking about real-world evidence for post-approval safety and effectiveness even, and think about when real-world evidence can supplement or even sometimes replace a randomized trial. So some examples are providing an external control arm for a rare disease, in some cases where a randomized controlled trial is not even possible. It could be used to augment clinical trials with additional subgroups or additional external comparator. It can be used to conduct an active comparator study, and sometimes it could replace post-approval trials like large cardiovascular outcomes trials, or be used to support label expansion for a product that's already on the market. But in all of these cases, we're really thinking about the evidence here as something that's going to support an important decision. And whether that's a decision by regulator or an HTA, it needs to be decision grade. So how do we go from real world data to real world evidence? There are two main components. One is that we want fully traceable data, and the other is that we want transparent causal evidence generation. So what we mean is that from the very start of the data collection, through every step in its transformation until it becomes a research database and ultimately a cohort, that there are no black boxes, that the data are fully traceable. And we also want to ensure that we are using the best practices that allow us to conduct a valid study so that we can interpret our results as a causal effect. And we want every step of the decision-making process and the design and the analysis to be done and recorded so it's transparent and the decision makers can feel confident in the data that they're looking at. How do we do this? First, we follow the tenets of the Mvet real-world data framework. M stands for meaningful evidence. So we're talking about evidence that provides sufficient information and context to support interpretation and conclusions and decisions, that the evidence are valid and meet scientific and technical quality standards to allow those causal interpretations, that the data are expedited so that even if it's an incremental piece of evidence, it's helping facilitate timely decision making, and that it's transparent, as I said, that it's auditable, reproducible, and robust, and ultimately so that we engender trust in decision makers. And second, we need to use principled epidemiology. And in general, we like to think about our real world evidence in the context of what is the target trial that you would have conducted had you been able to. If it was feasible, whether that is logistically feasible because you could have the patients or you have the time and the money to conduct an RCT, what would that RCT look like for this particular research question? And there are a lot of components here, making sure that you have the right research question, making sure you have the right patient population, that you identify and adjust for confounding, that you have the right treatment and comparator, and that you can measure outcomes in a valid way, and that you conduct your analyses correctly, and that you use sensitivity analysis to increase confidence in your results, and then finally that you report out your results in that transparent manner. So that's great. Those are the two tenets, but how do we do this? One way is to apply the SPACE framework, and SPACE stands for a structured pre-approval and post-approval comparative study design framework to generate valid and transparent real-world evidence for regulatory decisions. And everything in this paper that I published with two of my former colleagues last year also would apply to any other major decision maker like an HTA. So the SPACE framework builds on existing guidance and frameworks, including that Mvet framework that I went over a couple of moments ago. It provides a process to identify design elements and minimal criteria for the feasibility and data concerns and validity concerns for documenting decisions. It starts with articulating the research question in a very clear way so that you can identify the components of that target trial that you would have done. And then you're making very conscious decisions about where to build in more pragmatic choices to replace that target RCT. And helps you identify areas where you need to use sensitivity analysis to support your design or your decisions and your interpretation at the end. So there are four major steps in SPACE. The first is to articulate your study design. And the second is to plan your model, really think through what confounders are going to arise in your study and figure out how you're going to measure them and adjust for them. It has a step of identifying the data elements that would be needed to have a valid study, that then is a necessary step for doing feasibility assessment of data sources. And then ultimately, you capture your validity concerns and solutions and your ultimate design. So starting with the study design, this includes 4 steps: articulating the research question, describing the design elements in terms of study population, treatment and comparator groups, outcome and length of follow-up, conceptualizing that target RCT to capture those elements of 1B, and then to think through where you need to build in pragmatic elements into your design. And this is an example. This is showing the table 1 from the space paper. And the box in green is highlighting where you'd record in this table those steps from 1A, the research question, down to 1D. So the next step is model planning. And here, ideally, you would draw a causal diagram for your treatment and outcome relationship of interest, and that would apply to any primary treatment outcome relationship specified in the study. So if you had one treatment comparator relationship that you were looking at, but with 2 primary outcomes, you'd have to do this twice. And then drawing out that diagram helps to identify potential confounders, and then we generally would want to look to the literature and try to understand the strength of evidence for each of the relationships between any potential confounder and the treatments and any potential confounder and the outcome, and make sure that the literature supports the causal diagram that we've created. And that is done in space in step 2, and actually takes you to another table that I'm not going to show in the interest of time. And then the next step is to specify your requirements to validly capture all design elements. And that's done here in step 3. So an example of this would be in the study population, when you're describing your inclusion-exclusion criteria, if there was a critical criterion needed, that you needed a, let's say, a particular molecular status for patients with breast cancer, you would need to make sure that you had a data source that captured that molecular status for every single patient. Otherwise, you can't apply the inclusion-exclusion criteria that are critical to your study design. And so that's an example of what you'd capture here in step 3. Once you have those requirements specified for all the design elements in step 1, then you can conduct a systematic feasibility assessment. And usually I would advise that somebody starts with identifying data sources that have the sufficient number of patients that meet the inclusion-exclusion criteria, because that very often narrows the field of data sources as quickly as possible, so that then you're doing a more detailed assessment against only those data sources that have sufficient sample size. And then evaluating each of those data sources against the key elements. And this is a very involved process and is a step beyond space. But this would include something like making sure that not only does that data source record all the key data elements, but also what is the extent of missingness in those data elements, and do you have all of your treatments, your inclusion-exclusion criteria, your treatments, and your outcomes of interest recorded for most patients. And at this step, in addition, when you're looking at various data sources, you can consider if there are more than one data source meets the needs of the study, combining those in cases of rare exposures and or rare outcomes. So part of that process is to think about the data relevancy, making sure that the data have all the key elements that you need, that it's representative of the patients that you actually wanted to study, has sufficient sample size, the longitudinality is okay for your research question, and that can include time going back before the patient is diagnosed and treated and time after, allowing for sufficient follow-up, but also looking at the data quality. So maybe it has all the components, but are they fully captured? Are they valid? Where did the data come from? What are all the hands it's passed through, and can we trace it back? And then finally, we want to capture the validity concerns and solutions as we're going through the process of thinking through our study design. So that would be done here in step 4. And there's some tools that you can use. You know, some of this you may record while you're doing your feasibility assessment. So you may find a data source that looks pretty good and it has fairly complete data, but maybe one of the key confounders is missing for 30% of the patients. And that's something that you might want to record as a validity concern here in step 4. But a lot of this will be captured as you're thinking through this flow chart. So this flow chart is also from the SPACE paper, and it takes you through several steps, thinking through whether randomization is feasible. If randomization is not feasible, like we're now looking at the right-hand side of the flow chart, we're asking if there's a data source that does meet the requirements that we specified. If the answer is yes, then we can consider using a secondary data, that secondary data to do an observational study. And then we have to think very carefully about whether we can control for confounding. And if the answer is no, then you're sort of in this red box here on the lower right-hand side. And we would basically be recommending that you don't do an observational study, and then maybe revisit the research question and go through the process again and see if you can end up in a different end place in this flow chart. But there are two boxes here that would allow you to conduct the study, one using the secondary data, and then the other one on the far right-hand side using a primary data source. So once you've gone through this process, you essentially know what kind of study you're doing. Are you doing an observational study or something with baseline randomization? And you know whether you're using a secondary data source or a primary data source. And that can be recorded back on the table from SPACE. But this is an example of a tool from another paper from Jesse Franklin that was also published last year on evaluating the use of RWE for regulatory decision making. And I find that this is a very helpful tool to use side by side with SPACE as you're going through that flow chart, as you're thinking through the validity concerns, to make sure that you haven't missed anything. Just sort of a reminder checklist to use. So ultimately you have a final decision and you can record that here. And then you're ready to really design your study, get down to writing a protocol and then ultimately implementing your study. So how do we do this? The first thing that we recommend in terms of best practice is to describe your study visually. This simplifies review and communication. It encourages researchers to think systematically and also can ease the identification of potential errors. So I'm going to show a quick example. I'm not going to read this to you, but I'll let you just glance at it. This is from a paper published in 2011. It's actually a quite thorough study description, but it's obviously text heavy. I'm going to highlight some of the pertinent points that are in this paragraph. Talks about when cohort entry was assigned, who the patient population is. These are people 65 and older, that they had pharmacy dispensing between 365 and 485 days before cohort entry, etc. So it's not so easy to pull out the pieces without the highlights. The visual version would look like this. So those highlighted pieces of information appear in bullets on the left-hand side here, but all of this information is shown visually in this diagram. So we can see that we had exclusion assessment windows on cohort entry on day 0. We had that were related to age. We had exclusion assessment windows around the pharmacy claim that started well before cohort entry. We had an exclusion assessment window here that went back a year and ended the day before cohort entry for death. And then the covariate assessment window here is labeled as the year before cohort entry. And then follow-up here is defined as cohort entry going forward for 1 year. And all of that can be seen very clearly in the diagram. So these diagrams also would allow us to identify errors. So if you were assessing someone else's study and they had this diagram, then you would see immediately that they have an issue where they're assessing covariates, not just in the baseline period before cohort entry, but it overlaps cohort entry and follow-up. And that means that there could be some covariates that are impacted by the treatment, and you now have a temporality issue, and you could be adjusting for intermediates of the treatment. Another example is, in this example, we're showing the potential for immortal time bias. So we have an exposure assessment window that's overlapping with the follow-up assessment window, which can allow someone to have some person time prior to treatment that we have to then account for properly in our analysis. So the next thing that we should do when we're designing our studies is think about sensitivity analyses at every stage. So this really isn't typically thought of as sensitivity analysis, but it essentially is. When we do pilot work or exploratory analyses to help us design the best, most valid study, that is a type of sensitivity analysis. So before you even start looking at the relationship between your treatments and your outcomes, or your exposure and your disease, there are several things you can do to help inform your design. You can look at your data and make sure that you have a representative study population. You can evaluate the exposure and outcome definitions and windows. You can do different analyses to, if you have a treatment of interest, you may try multiple comparators until you find the one where you're able to achieve baseline balance. You can make sure that you have sufficient look back for covariates and new users and incident outcomes, and try different look back periods. And then you can evaluate the completeness of your exposure or treatment information, your the outcomes of interest, and your important covariates. So let's just look at one hypothetical example. So the hypothesis here is that a full course of antibiotic A reduces all-cause mortality compared with other antibiotics. And you can see here, this is an example of a directed acyclic graph, which lays out the relationship between antibiotic A and mortality, and shows all of the variables that we would need to consider as we're thinking through controlling for confounding in this study. So in this example, a regulatory body has asked for a replication study to confirm a prior finding. And the previous studies used comparator C, but there was some concern that the comparison between antibiotic A and antibiotic C, residual confounding may have resulted, it may have explained some of the findings. So during the design phase for the new study, the researchers used propensity score matching with 3 possible comparators to identify the best antibiotic comparator that was able to achieve balancing, that looked like pseudo randomization at baseline, and found that covariate balance was insufficient for comparator C and comparator D, but sufficient for comparator B. They provide that evidence to the regulator, and the regulator agreed that comparator B was the best comparator for the study. And this is all done prior to looking at antibiotic A against mortality in the data source for the study. The next place we can use sensitivity analysis is for a priori or pre-specified sensitivity analyses to occur at the time of analysis. And so this would be built into the protocol or the statistical analysis plan as you design your study. So this could include alternatives to the exposure and outcome definitions and windows, subgroup analyses, alternative comparators, adjudicated versus unadjudicated outcomes, imputation for missing data versus no imputation, alternative confounding control methods, and there are others. This is just an example, but the point here is that these are sensitivity analyses that you plan in advance and you deem important. And I generally like to think of sensitivity analysis as something that you might have made a decision for your study because it allowed a larger sample size and you thought you'd be more likely to be able to test your hypothesis, but there is a perhaps more valid way to do it where you'd have a reduced sample size. To me, that's an ideal circumstance for a sensitivity analysis because you're increasing validity and you're really focusing on what that effect estimate looks like under the more valid, but maybe with insufficient sample size. You're really then comparing the effect estimate against the primary analysis. So here's a hypothetical example of a pre-specified sensitivity analysis. So again, same example, we're looking at whether a full course of antibiotic A reduces all-cause mortality. And during the development of the statistical analysis plan, 2 sensitivity analyses were pre-specified. First, they stratified by infection type, and this is an example of what I was just speaking to, that the stratification they did not think would be sufficiently powered, but it was potentially important to look within the infection types that the antibiotic is used for. And ultimately, they found that there was a 50% reduction in mortality among those with upper respiratory infection indications, and that was really driving the protective effect. Another sensitivity analysis that was pre-specified was to use inverse probability weighting adjustment versus propensity score matching, contingent on there being no positivity violations. And while the regulators wanted propensity score matching because this was in line with what other similar studies had done previously, IPW was performed and resulted in similar findings, which is just adding to the evidence base and providing comfort that the results are robust. And then finally, we can have post hoc sensitivity analyses. And these are sensitivity analyses that you really can't expect or plan for. You have to wait to see what happens when you actually start analyzing your data and doing the analyses that you planned. So an example could be potential impact of unknown or unmeasured confounding. There's always the potential for unmeasured confounding in a purely observational study, but we can't really do a sensitivity analysis on that until we have our results. We could also look at the potential impact of imperfect measurement or follow-up if that's discovered through the conduct of the study. And then there may be subgroup analyses. If you had an unexpected finding and you're trying to dig into it and understand what's driving it, you may want to do subgroup analyses, for instance. So just one more hypothetical example, using the same example. During this pre-specified subgroup analysis, they identified an unexpected finding. So in the example, among those using antibiotics prophylactically, a 2-fold increased risk of cardiovascular mortality was seen for antibiotic A. That was an unexpected finding. So a post hoc sensitivity analysis found that imperfectly controlled confounding might have resulted in the finding. So they added a sensitivity analysis for the primary and secondary analyses, removing prophylactic users, and found a stronger protective effect. So essentially, the inclusion of people using antibiotics prophylactically was actually reducing the cardiovascular benefit of antibiotic A in the overall analysis. So before I wrap up, I just wanted to mention that there are benefits to using a platform, and there are multiple platforms out there, but it's very important to make sure that it's decision grade. And the benefits of a platform that is built with causal inference in mind is that you can make sure that you have meaningful data, meaningful research question, that your data are fit for purpose, and that's all done and evaluated through the platform. It helps you make sure you have valid data, so allows for methods that are best practices and scientifically validated, to have structured workflows that can balance flexibility with guardrails to make sure that you're meeting the assumptions of each analysis type, can allow meta-analysis and things like high-dimensional propensity scoring. A key benefit of a platform is expedited evidence. So being able to do something in real time, rapid cycle, to turn around data when needed for decision makers, taking advantage of libraries that have pre-built measures or algorithms for different diseases that may comprise the indicated population or the outcomes of interest. And these can be based on validated algorithms and are quickly reusable. And then importantly, the use of a platform allows for much more transparent research. There really are no black boxes. You can see from data ingestion and any transformation that is done to the data once it's in the platform, to identifying the cohort looking at subgroups, seeing how measures are built, and then ultimately exactly how the analyses are done and the ability to replicate the analyses or change them to do your own sensitivity analysis. So in summary, producing real-world evidence from real-world data uses or requires a principled process. We need to make sure that our real-world data are meaningful and timely, transparent, and ultimately trustworthy. And we think the best ways to do this are to imagine the target trial that you would have conducted, adding pragmatic elements as needed to make the study feasible, and then using the existing frameworks and tools such as Mvet and space and the design visualization. And ultimately, when we have more prescriptive guidance, making sure to meet the needs of the guidance, and conducting systematic and well-documented data feasibilities is really critical part of the process. And then using sensitivity analysis throughout the study lifecycle to support your evidence. So as someone who is looking at a final study report and trying to evaluate the evidence, if the researchers have used these tools and they're providing, for instance, that table from space and it's completed, you can trace back and see exactly why they made the decisions they made. If you were expecting a particular confounder to be controlled and ultimately they didn't include it in their propensity score model, you should be able to see in that documentation exactly why they made that decision. Okay, I will stop there and we can open it up for Q&A.
Thank you, everyone. Thank you, Nicole. Hopefully you guys can hear me. I know that there were some technical difficulties for a few folks, so hopefully we've gotten those resolved. Liz, are you there with me?
I am, yes, hello.
Oh, great. Hey, Liz. All right, so I know that we have a few questions coming in. My slides disappeared, didn't they?
Yes, you're showing your desktop.
All right, I'm working on that. All right. So while I kind of jump to the correct slide again, so we've had a few questions come in. In the meantime, while I'm trying to get my slide up, one of the questions, Liz, is around unmeasured confounding. And can you speak to kind of how how researchers and HTA folks can think about unmeasured confounding when they're either generating real world evidence or evaluating that real world evidence?
Well, the thing about unmeasured confounding is you're not actually able to predict it fully. There's always going to be a concern for unmeasured confounding, but by having sensitivity analyses, and we're going to talk a lot more about sensitivity analyses tomorrow, having numerous sensitivities that evaluate the robustness of your findings help to understand whether or not unmeasured confounding is a large concern on whether or not it can be fully minimized. I don't think it can ever fully be eliminated, but we do everything possible to minimize the potential for unmeasured confounding.
Yeah, great. And then another question was around, we talked at a very high level, or Nicole talked about a very high level of, for example, creating good balance between the intervention group and the control group, or the two groups that you're looking at in your real world evidence study. Do we have any thoughts or guidance around what actually good looks like? What is sufficient? What is good versus bad versus great?
Right. So what we typically would consider as a rule of thumb is an absolute standardized difference less than 10% to identify a lack of imbalance. Typically, if you see absolute standardized differences that are greater than 10%, you might have some concern for residual imbalance even after your methods have been applied. And that kind of links to the prior question also, in that if you're establishing balance across between your two comparisons, that you're getting closer and closer to emulating the randomization of a trial, and you're also getting closer and closer to minimizing that potential of unmeasured confounding. So the assumption is that you're potentially achieving balance even in the things that you haven't accounted for.
Yeah. And another question that has come in is, as an HTA body, what are kind of some of our recommendations for understanding and articulating and setting those standards for what is sufficient? So like you're saying, that 10% rule, you know, what types of activities can HTAs potentially engage in to really help them understand what good luck looks like for, you know, their particular use case?
So I think, you know, a lot of the things that Nicole outlined, there's a lot of different metrics that a lot of, you know, people have put a lot of thought into this in the real world community and have put together a lot of really good checklists that HTAs can follow and adapt accordingly. And I think following those types of checklists, as well as, you know, trying as best as possible when it is possible to get your hands dirty and to consider additional sensitivity analyses, I think that's really how you can kind of hone in on what is good. I think at the end of the day, there's still some subjectivity about what is good, but if you've kind of satisfied all the checklists, you're getting very close to good.
Yeah, and I would also add, you know, like you said, there's a lot of research, there's a lot of activity that has been done around kind of setting some standards for the building blocks of RWE, but I still think there are some unknown questions. And for example, the FDA is doing demonstration projects to really kind of zone in on situations where RWE will be useful or will not be useful, what types of questions it can and cannot answer. And I think one of those good examples is RCT duplicate. And without kind of going down the path of, you know, very focused on regulatory, I think it's just a good example of kind of some of the activities that maybe HTAs could engage in, kind of adapting specific questions and designing research projects around those specific questions. So can you talk just a tiny bit about the RCT duplicate project within that context?
Right. And so I think one of the biggest things I think is important to consider for RCT duplicate is that it's not just about the expectation of being able to emulate all trials, because identifying certain types of trials that aren't easy to emulate via real world data is also considered a success of the project, is identifying certain trends. Like, for example, you know, a lot of studies that are using real world evidence are using commercial claims or, you know, federal claims like CMS. And those types of databases don't have cancer staging. And so if you have a major cancer outcome, you need to have a data source that has the cancer staging and the adequate information available.
Yeah. We did get another question around data collection. So the specific question is, how can we as HTA methodologists collect real-world evidence in developing countries where real-world data is really not collected in a systematic or accurate manner? And are those situations where we should be just only using prospective studies? So I think it's kind of two questions. One, you know, using prospective versus retrospective studies, and as kind of a community, how can we help set standards around RWD data collection?
That's an interesting question. I mean, I think, you know, there's a little bit two different schools of thought on whether prospective data collection is considered real world data. I guess some would argue that it depends if it's collected in more of a real world setting, then it would be considered real world data as opposed to, I guess, non-real world data. But I think it's really about analyzing the data over the actual data collection process, regardless of how it's collected, whether we're using robust methodology to analyze the data and to fully account for, various baseline comorbidities, for example.
Mm-hmm. Great. Another question has come in around that residual confounding. So isn't the best way to minimize? So isn't it the best way to minimize residual confounding, selecting your population to ensure that individuals would be candidates for either the intervention or the comparator? Even the best propensity score matching approach could fall apart in the presence of channeling bias, for example. Any thoughts on that, Liz?
I'm sorry, I got a little bit lost in the question.
I think the question is kind of a comment on minimizing residual confounding and kind of trying to ensure that balance. Is there ways to select your control group or your RWE comparator groups in a way where you can kind of minimize that, the chances for confounding?
Yeah, and I think calendar time bias is a really important concern, and that's why thinking about external control arms, there's really a shift away from what had historically been called historical control arms, and I think it really depends on what your definition of historical is. You don't want to be using old data as a comparison to new data. What had happened in the past is sometimes you have a new drug and you are comparing it to an older standard of care. And I think we're always going to be up against some of those issues, especially thinking about external control arms. But I think it's important to understand how the medical practice has changed during those dates and to really restrict any older standard of care data to the most recent possible to minimize the potential for all that channeling.
Yeah, and kind of on that topic of external control arms, I think one of the questions that we've been getting lately is around international data. So, for example, if a randomized control trial is ran in the US, how applicable is that data to UK, for example? Or if my real world data is collected in the US, how applicable is it to outside, ex-US HTA bodies or regulatory bodies? And I think that, this idea of trying to match time with your external control arm, trying to get, the closest time frame for your intervention group versus your control group is really great. But I think there's a lot of other considerations around using international data around ensuring that the standards of care are similar, the types of care are similar, the different delivery mechanisms are similar. So one of the questions is around kind of international data. Any thoughts on other than kind of doing your research on kind of the historical context and the treatment pathways within the country? Any other thoughts on how we can try to ensure that if we're using an external control arm, it's applicable to the country of interest?
Well, I think that's I don't think there's an overarching way to answer that question, because I think it really depends on the research question of interest. For example, issues about mental health, for example, might be very different in the US than Asian countries, for example, where, there's still a lot more stigma around mental health. But for things like, COPD, I think a lot of the same drugs are being used in care in Asia as they are in the US. And I'm just using Asia as an example, but I think it really is going to depend on the research question, and it's important for any project or proposal to have a full assessment of the data fit for purpose. And the data fit for purpose also includes those generalizability concerns.
Yep. Yeah. Yeah. All right. Another question is around, as an HTA reviewer, if you're receiving information from the manufacturer, and even if they're kind of following the space process, you could potentially follow the space process, let's just say 3 different ways, right? Like way 1, way 2, way 3, and kind of choose, cherry pick the results. And that's one of the kind of critiques of real world data, is this ability to kind of cherry pick. Can you speak about kind of the, transparency issues around ensuring that that kind of qualm with RWE isn't happening, and how to safeguard against that?
Right, and I think that's an important concern. And I mean, all regulatory submissions require documentation of not just the results, but how you got to the results, and documenting your path of where you started, having an a priori plan in your protocol, and then how you got to your results is really key to transparency in real world data, real world evidence research rather. And I think I think we'll talk a little bit more about this tomorrow also, but like use of automated platforms that track each iteration is something that is a valuable tool to have, because then even if you do have to, change a covariate rerun, things like that, and there are justifiable reasons for that, that you're not just highlighting the end game. You have this whole trail of where you started and where you ended up.
Yeah, and another thing with the, I think it's ISPOR, I think it's both ISPOR, ICBE, the real world evidence transparency initiative. One of the things that they're advocating for is the pre-registration of the study on other, clinicaltrials.gov, or yeah, exactly. So that could definitely be another way to safeguard against this idea of
right, because that should be all methods that are established a priori before actually implementing the analysis. But even if there are post hoc changes that are made along the way, which does happen, especially with real world data, because you realize that the way that the data is captured for a certain metric, you need to modify accordingly. Then as long as those decisions are justifiable and documented along the way, I think you still have that full transparency.
Yeah, and probably another point is this idea of collaboration with the regulators or with the HTAs. If you go to them early and say, this is what we're thinking about doing, and this is why, opening up that dialogue very early. This is what we tell a lot of our clients. Opening up that dialogue early is a lot better than just kind of plopping something on their desk at the very end.
Right.
So that can, again, kind of help with the transparency issue.
Right.
Yeah. Great. Well, I think those are the questions that we have. The next thing that we wanted to do is really preview what's going to happen tomorrow. But if you had technical difficulties, or if your question didn't get answered, you can please feel free to email me any questions that you have, either right after the presentation or when you rewatch the presentation sometime for the rest of the year. All right, so in terms of what we're going to be doing tomorrow, hopefully you will be able to join us. I think it's going to be a bit more interactive than today's kind of more lecture format. But the idea for tomorrow is to choose your own RWE adventure. So we're going to take a hypothetical HTA submission. So our hypothetical case study will be a manufacturer generating an RWE comparative effectiveness study to kind of prove or demonstrate why SGLT2s should be a preferred second-line therapy over GLP-1s. And we're going to take that hypothetical example. We're going to walk through the real-world evidence, how it was generated, what the results were, and then we're going to apply what we learned today, and we're going to try to evaluate the quality of that submitted RWE study. And we're going to do that by pressure testing the results. And so we're going to flash you a few different options of routes that we would want to take in pressure testing the results. And we're going to choose, which route is most popular. And then we're going to take that route and look at the results of that kind of sensitivity analysis. And then we're going to sum up kind of what we think, after that pressure testing, we're going to sum up kind of what the main takeaways are and try to get to a point where we can either say yes or no as to should SGLT2s, if they should be a preferred second line therapy. And again, this whole idea is really to start to learn and start to understand how we can evaluate RWE studies and if they're providing sufficient evidence for us as a HTA body, for example, to make a decision. So that's what's coming tomorrow. Hopefully you will be able to join us. If not, the session, I believe, will be recorded and again will be available after the fact, but you won't get to vote, and so you won't have any say in which route we choose. So hopefully we'll see you tomorrow. So thank you very much for your attention today. If you, again, have any questions, please feel free to email us. Thank you.
Thank you.